Okay, hi everyone, welcome back. So the first speaker of the session is Hiroaki from the Polish Academy of Science, and he's going to be talking about functional validation of SNPs.
Okay, good morning, everybody, and thank you very much for inviting myself to be a BOBREC meeting in Brussels. And today I'm going to talk about functional validation of SNPs in specific trait by well, I cannot say, utilizing molecular tools, right? So it would be better because my eyes are quite bad and I cannot read this one. So genomic data allows to identify regulatory elements and SNPs in key tissues of different livestock species, including cow. So the major question that we have is how regulatory elements and SNPs influence gene expressions and subsequently modulate phenotype. And then this is the topic and task that we worked on work package 6. So a little bit of strategy. So basically, we took the data coming from GWAS analysis, which is done in work package 2. And then also like chip sequencing, attack sequencing that is coming from different work package. Like GWAS analysis is from work package 4 actually. And chip sec, attack sec coming from work package 2. And in different work packages, like we in work package 6, we first did the prediction of regulatory SNPs and enhancers in the work package 6.1, and then 6.2 like did the luciferase reporter assay based screening, and after that in work package 6.3 we did the CRISPR-mediated gene editing for functional validation. So a little bit more detail. I don't have time to talk about all the data done in our work package, but as for regulatory SNPs, we took existing NGS data and then using GWAS analysis coming from beef and dairy treat, and together with RNA sequencing, and attack-seq data. And Dominique and Matthew from India worked on prediction of regulatory SNP element using established software by them. And we kind of identified putative 31 different regulatory SNPs. And afterward, we did the promoter assays using luciferase assay. And then finally, we identified 3 different significant regulatory SNPs. And then now we are in the middle of we are trying to finish the functional validation of these elements, like using CRISPR-mediated gene editing. So today I'm not going to talk about promoter part, but mainly like focus on enhancer part. Like the same is true for enhancer. We did the beef trait GWAS and RNA-seq and attack-seq data from work package 4 and 2. And then we identified 4 putative enhancer element, and then using a reporter assay, we kind of like narrow down the target regions in 3 different regions, and then perform CRISPR-mediated gene editing again and RNA sequencing. So shortly, like how we do the reporter assay, basically like we target like enhancer region and promoter region, and took these regions and put it in plasmids, and then like transfect it like into the cells, and then perform the luciferase assay using some specific substrate. And then in the case of SNPs, like you mutate the target element and then compare with the original allele. So this is the data that we did for the enhancer screening. So since the data is not published, like we just name it like enhancer 1 to 4. So as I mentioned, like only enhancers 1 and 2 and 3 are having enhancer activity, as you can see here. And since enhancer 1 had 13 SNPs in a very narrowed down narrow element, we didn't do like mutagenesis, but instead we took the different approach that I'm talking about later. And as for enhancer 2 and 3, like when the mutations are made like for specific nucleotide, we had reduced luciferase activity, which shows the some of the area is like specific significantly changing the enhancer activity. So as for enhancer 1, we are quite interested in working on this because in a very narrowed down element which has like 500 base pair, we had identified 13 SNPs like coming from beef trait that is done by GWAS analysis. And then what we did is like simply we used like 2 different CRISPR guide RNAs, as you can see here, and then cut off this region to see the effect of it. So as you can see here clearly, if you look at wild type, like when you do PCR, you have like 100, 500 base pair of the amplification, whereas like CRISPR clone has shorter amplification just because like we deleted this element completely. And the result is the PCR product is like directly sequenced, and then we confirm the deletion is properly made. So we performed afterward like RNA sequencing, and like simply like this is the PCA plot showing the wild type is quite distant from CRISPR clones. And we did the independent CRISPR-mediated gene editing, and we had the similar PCA plot like between different 2 clones. And we compared the gene expressions of 2 clones, like which lacks the SNPs accumulated element in enhancer 1, and identified 630 genes which are significantly changed as compared to wild type. And we found interestingly, like mainly downregulated genes are more than upregulated genes. And sometimes like we are asked like if the RNA sequencing data is like true. So we also performed the qPCR analysis. We took cDNA and then performed the qPCR for the significantly downregulated genes. Like as you can see here, like clone 1 and 2 had a significantly downregulated genes like for specific genes which are detected by RNA sequencing. And we performed also the pathway analysis, KEGG pathway analysis, and bubble chart. It's a little bit like difficult for the audience to see, but we found the cell growth and cell cycle and DNA replication pathways are changed, like when the deletions deletions are made. So we are currently thinking to do further validation, function validation, if when the deletion is made, like if the cell cycle or cell growth is changed in MDBK cells that we use for this study. And we also worked on high C and 3C assays to know the chromosomal interaction with specific interaction between like this enhancer and BIN1, because in work package 1, like we identified this enhancer 1 is highly connected or interacting with BIN1. So this is like kind of methodology, how we do the 3C. I'm not gonna go for in detail, but basically you cross-link the cells and then like we enzyme digest the interaction and then put the ligase to ligate the interaction, and after that you do high C, like sequencing or 3C based PCR to identify the interaction. And we confirmed more than 10 interactions in terms of this enhancer 1 and then specific gene element named BIN1, which is related to myogenesis in mice, like if the region is deleted, it causes myopathy. So it's something to do with muscle. And we also performed the interaction change. I mean, we validated the interaction change between normal condition and stress condition. And unfortunately to say, or I mean, like, it's hard to expect like what to see, but we couldn't see the quite difference between 37° and 41° when the stress is added. And also, we were kind of expecting to see if this deletion is made if BIN1 gene expression is like changed, and we were hoping that like this gene expression could be reduced. However, we didn't see the reduced expression in terms of BIN1 expression like when the deletion is made. So with the help of 3 institutes, including FBN, is the main contribution like for CRISPR gene editing, and URIM, University of Limoges, and us, Polish Academy of Sciences, we have generated more than 10 SNPs deletion mutants, which we didn't have time to discuss about it in this talk, but we made more than 10 clones for CRISPR-mediated gene editing, as for Enhancer 1 study, we found that beef trait-specific SNPs in Enhancer element deletion showed 630 gene expression change, including cell cycle, specifically in cell cycle and growth pathway. And we have identified chromosomal interaction between Enhancer 1 and BIN gene in normal and stress condition. However, we didn't find any significant change between normal and stressed condition. And these are members like who are involved in work package 6, Caroline and Krista from FBN, and Dominique and Matthew from INRAE, and Veronique and Natalie from University of Limoges. With that, thank you very much, and if you have any questions.
Thanks a lot, Hiroaki. Any questions? I'll kick off, if that's all right. So you took the approach of, you take all your GRAS hits and you narrow it down to a relatively small number, and then you did your reporter assays on a relatively small number. Do you know what the relative merits are of doing like a massively parallel reporter assay first and not doing the prioritization and then kind of prioritizing afterwards versus what you did of the prioritization in silico first and then a small number?
You mean like how you filter, we filter out the number of the regulatory SNPs and everything? What, could you repeat that question again?
So yeah, would you be better off, do you think you're better off doing like a massively parallel reporter assay, so you don't really filter the variants first, you just test thousands of variants on gene expression? Or do you think it's more effective to filter in silico first and then just test a small number?
Especially like as for regulatory SNPs, like we try to see the transcription binding site. So I think it's better to, because our focus was like finding regulatory SNPs. So I think we had to filter out the transcription binding site. And I think the filtration was required for this purpose. Okay. And that was my answer.
And do you think it worked quite well at identifying the regulatory variants, the approach of checking the transcription factor binding site? So you had three had an effect, is that right?
Yeah.
Okay, okay, cool. Thanks. Any other questions? Oh. So is there a microphone? Sorry, if you just wait for a microphone.
Sorry, thank you. My question is about the model, the CRISPR cells you prepared. Do you think this type of protocol or preparation of models could be made on a systematic way to test any number or even
actually,
on demand?
This is a great question. We are currently working on that, and we submit a paper based on this methodology, and then the paper is after review, and then we revise the manuscript and resubmit the paper to our journal. So I think this could be like quite useful for bovine cell studies. Like a lot of things are done in different species, but there are limited number of papers like to show systematically to CRISPR the bovine cell lines, and this is going to be the first paper to show the systematically way of CRISPR-mediated DNA editing. And on top of that, we are not using plasmid, so which might cause sometimes the problem for the integration of the unnecessary fragment into the genome, but we use like synthesized guide RNA, so which could be like more safe to be used in the future study. Thank you.
Thank you very much. Any other questions? No? Okay. Thank you very much, Hiroaki.
Thank you.